Hello everyone, I am Iliana Serghiou and I am a PhD candidate at the Quadrium Institute. I am presenting my poster at the Microbiology Society Conference 2023. My poster is titled A Recipe for Success, Shotgun Metagenomics for the Skin Microbiome. So I'm going to start with an introduction to this research. A commonly used method to study skin microbial communities is amplicon sequencing. But this method is limited to short-read sequencing, targeted gene regions, low taxonomic resolution, and lack of microbial information on functional capacities. Because of these limitations, an emerging preferred method to study the skin microbiome is shotgun metagenomic sequencing. This is due to its ability to use short and long-read sequencing, and it produces high taxonomic resolution of all genetic material present and information on microbial functional capacities. However, the relatively low bioburden of skin makes obtaining enough DNA for shotgun metagenomic sequencing challenging. There are a limited number of commercialized kit protocols available that can produce high molecular weight DNA from skin in sufficient quantities that are suitable for shotgun metagenomic sequencing, but none have been specifically optimized to extract DNA from skin microbiome samples. So I aim to develop and validate an optimized high-throughput automated extraction method for recovery of microbial DNA from skin swabs that results in DNA with yield and molecular weight suitable for shotgun metagenomic sequencing using both the Illumina and Oxford Nanopore platforms. So I'm now going to briefly describe how I optimize this DNA extraction method under the section DNA extraction optimization. To optimize this method, I performed a set of experiments to compare different initial diluents and lysis procedures using overnight cultures. The finalized method incorporated an overnight heated lysis with lysozyme, a heated off-board lysis, a bead-beat lysis, and samples were then extracted on the automated Promega Maxwell. This method was then validated on adult volunteer and baby skin swabs. Parallel with the swab DNA extractions, I also cultured the skin swabs to look at the culture metagenomes. These cultures were then extracted using the same extraction method. The DNA was sequenced using shotgun metagenomic sequencing on the Illumina and Oxford Nanopore platforms to test the extraction method's ability to produce DNA suitable for short and long read sequencing. Data was then analysed using Kraken2 and Bracken, which maps reads to a reference microbial metagenomic database. All results that I'm showing now in the poster are for the finalised method validation on skin swabs. So moving to the results section for the swab overnight lysis incubation time. So the time of this overnight incubation lysis was key for yielding more DNA. I compared swab overnight lysis incubation times at 18, 20, and 22 hours, and I found that 20 and 22 hours of incubation was much more effective than 18 hours for obtaining higher DNA yields from skin swabs. DNA yielded at 20 to 22 hours ranged from 3 to 5 nanograms per microliter, and molecular weights range from 3,000 to 5,000 base pairs. So now moving over to the results section for species numbers recovered from skin. Here I found a good species recovery from samples sequenced at 5 gigabase pairs depths and subsample depths. So 5 gigabase pairs represents about 100% reads, which is shown here. There was a significant difference in species recovery from 5 gigabase pairs of data and 2 gigabase pairs of data and below. So 2 gigabase pairs of data represents about 40% reads, which is shown here. Now moving on to the results section for the shotgun metagenomic sequencing profiles from the skin swabs. These profiles show that swabs provide more information than culture profiles of the adult and baby skin microbiomes. But the nice thing to see is that the culture profiles confirm the microbiota in the skin swabs. These profiles also show that shotgun metagenomic sequencing on both platforms produced sufficient data for profiling and detected typical skin profiles for adults and babies. Overall, babies had less microbial diversity than adults. The adult and swab culture profiles generated are in the peach colour, shown here, and the baby culture and swab profiles are in the blue colour here. Now moving on to the results section for the comparison of skin swabs and cultures and comparison of the two platforms. So this analysis shows the total abundant species counts as a percentage between both platforms and the cultures and the skin swabs. Both platforms were comparable as identified very similar microbiota. Culture profiles also confirmed the microbiota in the swab profiles, but swabs identified more information. The adjoining lines show the abundant skin species that were detected by both platforms and between the swab and culture datasets, which is shown here in blue and here in purple. So finally, to conclude, an optimized DNA extraction method was successfully validated on skin swabs after testing multiple variables. This method is now being applied to a large sample set to study biological questions. Thank you all for listening, and I hope you enjoyed my poster presentation. I now have a quick interactive game for you to have some fun with. All you have to do is identify the correct skin microbiota images surrounding the human in the middle of my poster. There are printed names of these microbiota to the side of my poster display, which you will need to stick onto the images on the poster. There is a bonus question, which is to identify the transient skin microbe. Answers can be found on the reverse side of the instructions, which are located to the side of my poster display. Thanks again for listening, and hope to see you at my poster.